Hello again. So we got done with learning transcription and that we make RNA from it. However, there are three different RNAs that we're going to be spending some time talking about, particularly when we get to translation. All three are very important, and it doesn't happen without all three of these. They're all made the same way using the transcription system, but I wanted to go through each one, what they do, what's their job. So the first one we'll talk about is the one in the middle here, messenger RNA, because kind of what most people think about when you think about RNA is the message. Because the main idea of messenger RNA is it's taking the message of the DNA out of the nucleus and bringing it to the ribosome. We focused on that quite a bit last time. What is it copying? Well, it's copying the DNA of the gene, and it's going to then be transported out of the nucleus to the ribosome. Uh, One of the things we'll talk about next week is how we create this codon. So every three messenger RNA letters are going to make a codon. And then when we look at the codon chart, which I know most of you have seen before, you will see how they code for a particular amino acid, which is building blocks of protein, not DNA, but protein. Um, they're going to Before this messenger RNA, though, leaves the nucleus, it needs to be modified. So I do want to talk a little bit about the modifications that go on here. So one of the things that happens on the 5' end, we're going to get this methyl cap. That's going to attach to the 5' end. And on the 3' end is what's called a poly A tail. So we're just going to put a bunch of adenines in a row at the end. So it's referred to as poly A tail, meaning there's many A's. And basically, as this messenger RNA is floating around in the cytoplasm, there are nucleases floating around there that start chewing up the RNAs. Mostly its job is to get rid of viral RNAs and bacterial stuff. It has also a side effect in that it'll turn off um, messenger RNAs as well. So to slow that down, we'll put that poly A tail because it usually comes at the end and starts chewing those up one at a time. So the more A's you put on, the longer your messenger RNA is going to last. Another final step is it's going to remove introns. So there are these non-coding sequence in your gene called introns. They do not code for anything. A lot of this stuff is repeating sequences. A lot of people say that's where the viruses put their junk in there. Uh, we kind of just basically have these places in there to allow that junk to get in there so it doesn't mess up with the actual genes. So the exons are the actual part of the gene. So it'll remove these introns, it'll put these exons together, and then you'll have a functional messenger RNA transcript that can then leave the nucleus and make your protein when it hits the RNA. And we'll talk more about the codons and stuff next week when we actually work with the codon chart. Uh, all right, another thing, we're bringing these messenger RNAs to the ribosomes. Ribosomes are made up of two things, protein and ribosomal RNAs. That's one of the RNAs we need to talk about. If you guys remember your cells, you know that in the nucleolus, inside the nucleus, is an organelle called the nucleolus. That makes the ribosomes. So these ribosomal RNAs are made from the DNA. They travel to the nucleolus, combine with proteins to make the ribosome, which then leave the nucleus to go where they need to go. There's basically two subunits, aptly named the large subunit and the small subunit. And they basically attach to the messenger RNA. Messenger RNA will just kind of be attached to the center there. And it basically reads the code and puts in the right amino acids at the right time. So it kind of acts as an enzyme. So it's often referred to as a ribozyme. So remember, an enzyme is a catalyst. So basically it provides a platform for translation to occur and get the right amino acids in the right spot at the right time. and it gets them in close proximity, those two amino acids, so that they can form a bond and connect. Then I'll move over to the next one as well. So we'll talk more about that next week into translation, but then are on the run. The last RNA we need to talk about are the transfer RNAs, also called tRNAs. They have this weird cloverleaf shape here, and at this side we have the anticodon, so these three letters here will match up with the codons down here to bring in the right amino acid. This end here is going to attach the amino acid, so it's going to bring it in. And these two ends kind of act as a spacer so that it fits into the spots of the ribosome, as we'll see next week. There's two sites in the ribosome, and these spacers kind of put them in the right spots, the amino acids in the right spot, so that they form bonds. So if we were going to look at it, and we'll look more at this next week, we've got our messenger RNA code here. The first three letters are going to be the codon there. The anticodon matches it, so it's just the opposite side. So you've got an A, you're going to have a U, UA, because remember, both these are RNA. There's no T's. It's all the U's. And G is going to have the C. And then on the other side, we've got our amino acid, which in this case, AUG codes for the amino acid methionine, which is attached there. And that's going to be the first amino acid for all proteins. That basically ends my talk on the three RNAs. At this point, you should be taking the Google form quiz on messenger RNAs. Make sure you use your notes so that you know what's going on. That concludes the three RNAs notes.